Hello, my name is Sebastian Metz. I am a postdoc at the the laboratory of Lemar, and I will present you our research project that is called pathobiome characterization during the infection of Parkinson parasite in clam population.
My name is Aurélie Chambouvet and I am a CNRS researcher at in Lemar labs. And I'm a molecular ecologist working on parasitic protists, and my main goal is to describe and characterize emerging parasitic protists in aquatic ecosystems. So our lab focuses on one parasite, Perkensus sorsenai, which is an eukaryotic microparasite that has been co-introduced with its host, the Mania clam, from Asia through US. In European coastal area. And then, since the first introduction, the parasite spread along the Atlantic coastline with mortality events in Spain and in Portugal. So in a previous research project, we found out that finally there is not only one parasite, one host in the interaction, but there is a huge diversity of microorganisms that have been harbored in a clams population. So our main question was, what is the impact of this diversity on an exotic species like Perkensus sorsenai on the infectivity and the virulence? So we have two different cultures of two different parasites in the lab that we have sampled in our clams. So the first one, the pink one, is Perkensus sorsenai, the exotic parasite, which is this small cell here. And the yellowish is another parasite that we didn't expect to be there in Arcachon Bay. And in fact, it's Perkensus sesapiki.
Hello, I'm Philippe Soudan. I'm a researcher at CNRS, and I started my career actually working on the Perkensus, on his impact on the clams. We are interested on Perkensus is because it can kill clams or even oysters. It's a big important issue in Europe because the clams production is about 55,000 tons, so they can have a fairly high economical impact when some mortality outbreaks occurred. Most of the events occur in the south of Europe, Italy, Spain, Portugal. So why the south of Europe is more affected than the north is mostly because of the temperature. The Perkensus loves high warm temperature and high salinity, but then we can anticipate that this may go further north with global warming. But that's two things that we know are very important for the development of the disease in clams with Perkensus. But we try to understand what are the other biological parameters that can influence the dynamic and the evolution of the disease. And then we have to look also to the other microorganisms that are being hosted by the clams.
Like all animals, clams are not isolated organisms. They harbor millions of microbes that are called symbionts. However, under the influence of a pathogen, this community is named pathobiome. Recent studies suggest that by synergistic or antagonistic interaction, the pathobiome can play an important role during the infection process of the pathogen. As was explained by Aurélie, we have a clue that many other parasites are co-infecting the same clam with Perkensus sorsenai, and they may be related to the difference in infection intensity observed in Europe. Well, in this project, we will abort the hypothesis that the microbiome or the pathobiome play an important role in the infection process of an exotic parasite, such as Perkensus sorsenai in clam population. We will sample clams from different countries, Spain, Italy, Portugal, and France. The collected sample will be processed in the lab, where we will dissect the gills of the clams and extract the DNA. This DNA will be a pool of genetic information of the host, the parasite, and all the organisms part of the pathobiome. So we will use it to find out who is there and what are they doing. Using statistic and bioinformatics software, we will be able to decodify and elucidate the composition of the pathobiome, following by modeling the community observed in the different regions and the characterization of the host and the parasite. We will be able to get a better picture of the role of the pathobiome during the infection process of Parkinson's disease in plant population. This project has a lot of implications in many areas of interest. Among them, the fishery industries, diversity management or infection prevention. Since this parasite not only can infect Manila clam, but also another less valuable species, and they threaten with the population of bivalves in different areas that can have a lot of impact in the environment.